Go. Okay. Now, it's my absolute pleasure, like really, truly great pleasure, to welcome Sue Kidwell to launch our seminar series. So Sue is the William Rainey Harper Professor in Paleontology at the University of Chicago. And over the last few decades, really, I won't say how many, Susan's research program has made really groundbreaking contributions to our understanding of marine taphonomy, or more simply put, how should we read the fossil record? And she's really been also, really instrumental in the training and mentorship of countless paleontologists over the years. And more recently, she's been a leader in the novel field of what some people call conservation paleobiology. And there she took her the toolkit that she really developed in her earlier career, and she uses it to shine a light on young fossil records and how to interpret those young fossil records to understand how marine ecosystems have changed under human influence, which is, of course, exactly what interests our community. So today, Sue will give us a talk entitled Where We Are and What's Next in Understanding the Nature of Ordinary Shallow Marine Fossil Records. Thank you very much, Sue. We are thrilled to have you launch our seminar series. Over to you.
Oops. We practiced this multiple times, but I still bobbled it a little bit. Is that okay? You could see just the slide.
Looks great, Sue.
Okay, thanks. Hey, I was just so delighted to hear about the start of this project focused on shallow marine, and really honored to be asked to do the kickoff. Aaron told me I could go sort of narrow or could go big, so I went big, this just hilariously, hubristically big. But I thought, well, we might have a whole variety of different type of quaternary people here, and many of these things might be new. So I thought the idea of giving us a whole bunch of ideas to think about going forward at the start of your project might be fun. So I'm going to tell you a bit about what we do know, including what we know about the mechanisms, the dynamics and the mechanisms behind the nature of the fossil record, as well as things we still don't know and things we need to tackle. So I don't know about the rest of you, but when I think about quaternary, I think about these kinds of records, these sort of typically very finely resolved tree ring, ice core, whatever, beautiful pollen spectra, focused on a variety of different kinds of questions, but typically high resolution spatially and temporally. But what I want to talk about today is what I call the ordinary fossil record. And that is the kinds of material that accumulate on shallow subtidal seabeds with oxygenated overlying water. So this is ordinary rather than extraordinary exceptional preservation. So when we have oxygen present, that means we typically have no soft tissues. We've just got skeletal and other refractory debris. We find that it's largely from the fairly local within habitat community. Because of the overlying oxygen, these seabeds are subject to intense bioturbation. And that bioturbation with oxygen combined with organics creates a biogeochemically very complex surface mixed layer. Now, all of that is superimposed on, the geologist's perspective of a range of different net sedimentation rates, whether those are siliciclastics, which is what I work on, or carbonates. And on continental shelves, we're talking about a fraction of a centimeter per year on a lead-210 time scale. That is when you evaluate sedimentation rate on sort of a centennial to millennial time scale, up to a few centimeters per year in bays and deltas. Now, this is probably the most important slide in the whole deck, and many of you have seen this before. The most important thing I could tell anyone about the ordinary shallow marine fossil record is that the fossil assemblages are time averaged. And so this is my metaphor, the floor of a teenager's bedroom, where day by day they toss things off. A puppy comes in and removes something, a younger sibling comes in and drops a toy. But at the end of the week, you have what we would call a time averaged, or rather a time summed collection that captures the whole range of activities, and including rare events like a collared shirt because the kid had dinner with a grandparent. So when you sweep that together, it's a fundamentally different kind of picture of the inhabitant of this habitat than a snapshot of that teenager running out late for school. But it is can still be useful if that time window is not too coarse and if there has not been too much bias of the contents over that period of time. Now, stepping back again, we think about all the different qualities, all the different kinds of information we would like to have from fossil records, regardless of type. And they include a whole list, and I'm going to just briefly run through. For example, we care about what I call biochemical fidelity of the information that we might extract from the skeletal hard parts. By fidelity, I mean the faithfulness, the actual truthfulness of the information. We also care about its resolution, sort of the pixel size. That is the precision, the narrowness, the refinedness of the information. And we also care about the completeness, or all parts present. Anatomical fidelity would be also, sort of morphologic information. And we use all different kinds of methods to understand this, and I'll give you some examples of most of these, but we throw everything we have at this problem. Three other kinds of information that we want to evaluate, we want to use and thus evaluate the quality of, is the spatial fidelity, the spatial faithfulness of the biological information. And again, its resolving power. How much postmortem dispersal and blurring, spatial blurring of the information do we have? We also care about temporal resolution, that is the scale of time averaging. And of course, all the many things that can happen in that window of time averaged accumulation, including including the effects of having a longer window of time in which we sample the community. And then finally, what I call ecological fidelity, but this has to do with composition of the assemblage, whether we're talking about species or morphs or age classes. And that might be altered both by just time averaging alone, but also by active taphonomic bias, that is selective loss or addition of items in the window. And then finally, backing out, the completeness of a time series is every increment of elapsed time represented by fossil material, and what I call stratigraphic bias or mega bias, that is non-random selectivity in which facies get preserved in the stratigraphic record, secular variation in qualities of preservation, for example, the evolution of shell-crushing predators, those sorts of things. Okay, so today I want to really focus on this subset of qualities that we evaluate mostly using two approaches, that is live dead comparison and geologic age dating, mostly using radiocarbon of individual shells. And I'm also going to focus just on the material that we find, that we extract from the surface mix layer. We're also, Adam and I and many others now are working more on how that information gets captured during transit down into permanent burial. This is sort of, to me, the final scientific frontier of taphonomy, and when it's really difficult, impossible to approach experimentally. But that's a whole nother talk. Okay. So maybe the second most important thing I can tell you about understanding the nature of the shallow marine fossil record is that you absolutely have to think like a geologist. The first thing you have to do is you have to think about every single shell as a sedimentary grain, which is a terrible thing to tell a biologist, but you have to conceptualize it that way because that's what they are post-mortem. And that means thinking about them, the whole thing, from the source where they're produced and any transport to the sink and what happens there. And it's also the key thing that a geologist really does is that we zoom out, we stand back and we look at the entire landscape. We don't just focus at the shell in hand, and that having context, both temporally, but especially spatially, just helps with hypotheses, both creating hypotheses of possible origin and nature of the fossil assemblage, but also eliminating hypotheses. Okay, so some things that we've learned from live dead comparison. It's pretty self-explanatory how we do these things. Benthic realm, we take some kind of grab sample, usually by remote methods when we're working out in the continental shelf, but in shallower waters, you might use scuba or snorkel. And then we treat the live and the dead components of that sample as if they were the same, that is, counting species and comparing live and dead the way you might compare any two live samples. So some take homes from this, very early on, there were several just basic things we learned. And this is from the first meta-analysis I did. These are 85 data sets. And this is looking at just subtidal settings. So no washover fans, none of that kind of stuff. Subtidal level bottom settings. First thing we find is that typically, on average, there are 8 times more dead shell individuals than there are living individuals. That's the global median. And as you can see on that x-axis, sometimes we can have hundreds of dead relative to every sort of live. What that means is that in any given grab sample, no matter what the size, you quickly get a very large sample of the dead compared to the size of the sample of the living. In that sample, the dead typically has around 2.5 more species you would register than what you would register in the counterpart live sample, which means that using the dead, you can very rapidly accrue a regional scale species list. And then finally, what's shown there by the dashed lines is that you zero in where on the x-axis where you've got sort of 1-to-1 numbers of live and dead. So the sample sizes of the two assemblages are about equal. What you find then is typically you still have 20 to 30% more species in the death assemblage than in the living. And that's because the dead are distributed more evenly among species in any given sample, but also they are spread more widely across habitats than the live. So of course that could be taphonomic postmortem transport bias. Now, what we find, however, is zooming in, and this is where the work starts up with Adam Tomasovich, my beloved postdoc and long-term collaborator, is that these differences that we observe, even the most basic things about ratios of live dead ratios of individuals and species, and the increased evenness of the death assemblage, all of these differences can be produced, but the majority of these can be produced by time averaging alone. So this is the nice sort of this is a redrafting of a really nice sort of simulation. We analytically pooled living assemblages, short time series, over anywhere from 5 years and then through modeling up to 500 years to simulate how much additional richness would accrue through increasing the window of time. And what you find here, this is species richness on the y-axis, and at a local scale, a site or a single habitat, what you find is that species richness fundamentally sort of doubles. It also fundamentally doubles if you do the same thing, but now you're pooling all the data from across an entire landscape or seascape from a small region. You have the same sort of order of magnitude increase in richness just from increasing the window of time of observation. And these results are very comparable to observed live dead differences. And these are in these same 31 subtitle subsets, where if we compare the observed live richness with the observed dead richness, we see it's the same order. And clearly the dead are better at capturing rare species, and thus this true regional diversity. They also do a good job of capturing proportional relative abundances of those taxa, which is extremely hard for a biologist to get from any single survey. It's just too much work. The other thing we've learned, and this is another sort of meta-analysis I did, is that in fully natural settings, the live and dead compare very closely in species composition. They have a high degree of sharing of species between the two assemblages, including the rank abundance of those species. So the y-axis is taxonomic similarity. It's a sample size corrected measure called Jacquard-Chow. And we find that in settings that are pristine, sort of a steady state situation, we typically have more than 50% shared species, which doesn't sound all that good. But if you work with live data sets, you realize that 50% between any two live samplings would be pretty good. And then along the x-axis is the Spearman rank correlation. That is, and what we find is that that's positive and commonly pretty good. That meaning that the species that are most abundant dead are also highly ranked in the living assemblage in that habitat, and species that are rare dead are rare or absent in the living. Now, what you find if you go to areas where independent evidence, the people who worked there, and I go through looking for what kind of human stressors, you find that there have been some kind of human stressors in the system. It's in those areas and only in those kinds of areas where I find really poor live dead agreement using these coarse measures of taxonomic similarity and rank abundance. This is especially true. I looked at like 7 different stressors, but this is especially true. This shows you the results where you have anthropogenic eutrophication. Taxonomic similarity is lower. Typically, you have more species occurring only alive or only dead, showing that, some serious waxing and waning of populations. And you also, in terms of the structure, you find that the dead, and this is what really nails it, that it's nutrification, is that the dead is dominated by filter feeders that prefer sort of lithotrophic conditions that are rare in the living. And the live assemblage sampled now is dominated by organic loving and hypoxia tolerant species that are rare or absent in the dead. And so you can even get some flipping of the relative abundance. So when you get to this, because of the biological coherency of who is live only and who is dead only, this goes beyond a simple correlation, and we get increasing confidence that the live dead discordance we find here doesn't have to do with the dead failing to capture the living. It's because the dead, because its time average, remembers what the living used to be, and that human activities have shifted the composition of the living far off of its former steady state composition. And what I find is only settings with human stressors generate this scale of live dead discordance. You may be in an environment with high natural variability. No. Get good live dead agreement there because the death assemblage sums across all that natural variability. Okay, so to summarize, all this work, including Adam and I have looked at a lot of different things. Our major findings: under steady state conditions, dead faithfully reflect the living. And we can look at all kinds of different measures, including niche preferences, that is the bathymetric distribution of taxa. These are all consistent with the differences. The live dead differences in these settings are consistent with contrasting a snapshot with a time average sample. And so the dead are obviously then a very valuable complement and extension, seeing deeper into the past of the data you have on hand from the living. But where you find strong live dead discordance, it signals a recent change in the living, specifically an anthropogenic change in the living. It is not a problem of the failure of postmortem preservation. And so it's therefore quite credible then when we find live dead discordance to interpret the difference in terms of change that is outside historic range of variation. We can quantify its magnitude because we've got quantitative abundance data. We can parse the drivers that we can attribute which driver, because we know the biology of the dead only and the live only taxa. We can discern the dynamics of change if we date the ages of the dead only shells, find out lags and including in the and we can of course inform management and relevance, which is how we then segue into conservation paleobiology and the sorts of things that you're interested in. Okay. And she found that she had a couple sites where she did have some very low live dead agreement. And when she looked more closely, she found that this was because of some live only species. When she zoomed in, it turned out that those were new arrivers. They were invaders. And so these were previously unsuspected arrivers. This has been tested in a bunch of ways, well, here's another test. I like both of this. This is Andy Michelson working on ostracods in lakes on the same islands. Again, he found live dead mismatch in one of his many lakes, and it was the one that was associated with an agricultural pond back in colonial times. And I want to point out with both of these, this is site level data rather than habitat. And it's typical that you get this spread with site level data. And I think it's trying to tell us something about the biology of community, quote, degradation under stress. And that is that the community changes sort of little point by point. It's not the whole community degrades and loses its richness and changes its species composition, but that it goes bit by bit. And we see this over and over again. So I think the live dead is trying to tell us about that too. Finally, to segue into sort of climate, live dead abundance can be done. This is one of the first and few sort of studies of live dead to detect climate change. This is Eric Powell, really nice study, using existing commercial fisheries produced data on 2 commercial species of bivalves. And this is just presence absence data. And I love it. It's a map view, single taxon, as opposed to the whole community. And what he found is the orange points are where this taxon occurs both live and dead. But he had a bunch of sites, they're all shallow sites there in yellow, where this taxon occurred dead only. It's no longer living there. And then these light orange sites where it occurs alive only. It's apparently a new arriver, has not yet accrued so that you would regularly sample it in the death assemblage. Now, we need a lot more in terms of things we need, and particularly with this group, we need a lot more explicit tests of climate. Climate is a completely different sort of problem than these point sources that I've typically been working on so far of wastewater and bottom trawling, where you directly impact the benthos. Climate change is creeping, it's insidious. So what we're finding is we need proportional abundance data to detect live dead discordance and multivariate sort of display methods. The other thing I like to point out, and this comes from former students of mine who worked in the deep time record as well, and that is during times of known climate change, warming, what we find over and over again is that benthos are not responding to temperature alone, and sometimes they're not responding to temperature at all. We don't see southern species appearing, but rather they're changing to associated changes in productivity, in bottom currents, and in oxygen. And so we really need to break this simplistic assumption of temperature being important or dominant or even sort of present, and really rethink our niche modeling. Okay, here's an example we've actually just completed. We've got this in review. This is Caitlin Meadows from her dissertation work using a 15-year time series made available by Jackie Grebmeyer and a whole consortium working in the Arctic. This just zooms in on the North Bering Sea. There's a known ecosystem regime change. This is what the ecosystem scientists call it, associated with warming, retreat of sea ice, and a change, specifically an increase in the flux of organic carbon to the seabed. And these bar plots over here on the left show you the relative proportions at 3 different sites from the top to bottom, the relative proportions at each bar of different sort of guilds and the families within guilds in the live collected in the first 5 years, the middle 5 years, and the last 5 years of that time series. And you can see we have a documented decline in a change from dominance of the bivalve, the obligate deposit feeders, to dominance by facultative deposit feeders, as well as change in family dominance within guilds. And when we look at the dead, and the bar graph for that is shown over there on the far left, outlined in red, we find that the dead, in fact, correctly detect the change, shown in this pyramid correlation plot, when the dead are compared to the first 5-year bin of live data. And the second, they have much higher sort of similarity, and that the change, where the live dead discordance appears, is only with when this dead collected in 2014 is compared with the live collected at the same time. Okay, so now what do we know about time per assemblage? How completely and uniformly? We do this by age dating. We get an age frequency distribution. You've all seen these. The key thing from these is that we repeatedly find that we can have shells that are very old. It's one of the first studies down in Baja. Typically shells as old as the habitat, but that most shells are very young. And if we zoom in on just this first part, in fact, here the median age of all the shells is 50%. So information is not evenly distributed. It's not drawn evenly from all cohorts of shell input over the history of time average accumulation, but it strongly favors the most recent dead. We find this over and over and over again. There have now been a dozen or more sort of studies. These are just some that existed back in 2013 when I did this review. The median ages are always quite young, and we have these long tails of rare but old shells, and these can be up, to 12 and 20,000 years old. Fundamentally, we can have shells, arachnidic bivalve shells, surviving for as long as that seabed has been submerged subtidally. Now, why do we have these strongly L-shaped frequency distributions, little mechanistics? I'm going to keep this to one slide because I otherwise get obsessed. Adam and I find that the way this is not a single simple exponential function, but that when you have ordinary sedimentation rates and bioturbation, what happens is that initially the first cohort, there's abundant shells, and that's because they're newly input, but those shells, freshly dead shells, degrade very rapidly. You rapidly deplete individuals from that cohort from all different kinds of processes. And this is why they're residing in that upper surface mixed layer with aerobic decomposition that almost always creates acidic pore waters. Now, the few shells that manage not to die, not to disappear, attaining some kind of little micro environmental refuge within the surface mixed layer, or attaining some temporary sequestration by burial just beneath it, they start to degrade much slower. They have a longer half-life, and that's because they have a timeout from those horrible conditions, but also we're finding it's because they are digenetically stabilized. The microstructure shifts, and that's a whole other talk. That constitutes only around 1% of the total cohort. So when we're working on fossil records, we're working on the 1% of shells that survive those initially very high rates. And these shells become fundamentally eternal. These are the ones that then are 10 and 20,000 years old, and they can survive exhumation back up into the surface mixed layer, where we sample them and get these L-shaped age frequency distribution. Now, how and why is this? Adam and I launched a cruise. We have cores, but this is just looking at the surface mixed layer, which lead 210 tells us is the top 20 to 40 centimeters of the seabed. We looked at two sites in low net sedimentation, 0.2, and two sites with high net sedimentation. And notice on those frequency distributions that in the low net sedimentation rates, you get shells up to 10,000, whereas in the high net sedimentation rates, it's much shorter. It's an order of magnitude less time averaging in terms of the total sort of duration. And so the first order control on scale of time averaging is low net sedimentation rate. That is a low dilution of sediment of shells. And so this goes back to my earliest sort of stratigraphic work in the 80s. We also find we use the advantages of legacy DDT released from wastewater on this shelf in Southern California to also have a gradient in the depth of bioturbation of even modern fauna, and where we find, in fact, the lowest degree of bioturbation, the lowest reworking rates, we in fact have there the shortest scales of time averaging, underscoring the very important secondary factor of bioturbation. And then, there must be other things going on. We're also dating shells, Caitlin and I, up in the Arctic. A third order control, of course, is the saturation state of the overlying water. It's all oxygenated, but that just makes it even tougher. It doesn't increase the acidity of the pore water, but it means that acidity of the pore water, the shells dissolve and dissolve and dissolve, trying to buffer it, but there's always new low pH water coming in from the overlying. What we find is there's fundamentally no sequestration, very little sort of long-term persistence. These are results from just the first 200 shells. But what that means is that we've got a short, very acute, much higher temporal resolution, shorter memory, but on the other hand, high resolution. Okay, so those are what we know and how we know it, and some of the stuff, including the mechanisms of forming these shell marine records. Let me now show you one application. We're now, what we sort of find, all of us sort of in this field, but now what we do is we don't just do a live dead, we also do age dating, especially of the dead-only taxa, once we determine which ones those are. And then using the age information, we can unmix the time average assemblage to actually restore absolute time of occurrences. And this allows us to assess suspected stressors and unfortunately find new ones. And because the scale of time averaging can extend to 10 and 20,000 years old, although it's a very attenuated memory, this is obviously relevant to pre-industrial effects. I'm going to zoom in and show you one of my most fun sort of projects, again with Adam. In my whole career, this is one of my two most favorites, that we totally stumbled across. This Southern California shelf, there's Los Angeles, where because of the Clean Water Act, municipal wastewater agencies have been required by law to annually or semi-annually biomonitor all kinds of sort of benthic stations. And this has been going on for 50 years. And of course, there are a lot of oceanographic institutions in Southern California, like Scripps. And so this is, in fact, one of the best known continental shelves probably in the world. Now, what we find today's shelf, once you get below the shore face, right, below 30, which is around 30 meters in Southern California, it's muddy. It's everywhere muddy. It's muddy sand, it's sandy mud, and it's characterized by infauna, mostly burrowing clams, or mobile epifauna such as ophiuroids, and you see them here. And these burrowing clams, it's a very strongly detritus-based ecosystem, okay? What we find live-dead agreement is that we find the same species that are present alive are also present dead. So there's a good capture of this living detritivore. But in many, many, many samples, we also have abundant shells of filter-feeding epifauna that have almost no tolerance for soft mud, because these filter feeders, many of them attach permanently to the seabed. For example, pterobrachioid brachiopods, the Paleozoic, two different genera of scallops, calcareous worm tubes, barnacles. Paul Taylor has documented 37 different species of calcifying erect bryozoans. And these occur widely, not just a couple samples, and they can be 30 to 70% of shells, as you see here. And so this is just a total mismatch with mud. And Adam said, Sue, let's date these brachiopods. And since he'd been so great about working on bivalves all these years, when he really actually loves brachiopods, he said, absolutely. But we approached as geologists. We thought, hey, this is great. We're going to date the scale of time averaging in a marine transgressive lag, because that's what we thought this was. And the mud were the high stand muds, okay, that would have come out at some point in the last 3, 4, 5,000 years. So here's what we found. There's age frequency distribution, and you see, indeed, we had Liqueus, there's that cutie, all the way out. This is the middle shelf, and so it was submerged around 9,000 years ago, and in fact, we have shells going all the way back. But much to our surprise, they did not disappear several thousand years ago, but they came right up to the present. It's like, what's with that? They don't belong out there. So we looked in, we zoomed in, and then we found that, yeah, they came up into historic time, but they have been decreasing since the mid-1800s AD. And in fact, there are no shells younger than 1910. And we've since then dated an additional 100, on the order of additional 100 brachiopod shells. Adam just comes and vacuum cleans all of them out of every death assemblage I get. And now we found one specimen that died in 1940, but this is basically what this looks like. So this is what got us into sort of modeling the effects of varying the rate of input of shells and what that would do from an age frequency distribution. This is an application now of that modeling work by Adam, and it's a real tour de force. Now we have time on the x-axis, and that's 1500 AD, so Columbus, 1492, and there is today on the far left. And the gray bars are the observed frequency distribution, and the black line is Adam's reconstruction of the actual sort of population sizes over this time. And the difference, of course, is the 99% of shells that are lost. And giving sort of errors and uncertainties associated with the dating, we find that this community thrived for thousands of years. It declined, probably started in the 1820s, but not later than the 1870s, and was extirpated by the 20th century. So this was totally fabulous, but I realized we could not get it published where we should unless we explained why. So started looking, and of course, thought about the normal things like climate change, because Liqueus larvae do not like warm waters. But in Southern California, the warming started too late. It started in the 1920s in the subtitle based on core work by others. Thought, well, maybe it's because of all those nutrients from the wastewater, but that also started too late. These guys were toast by the 1920s. Siltation is clearly implicated. These things do not belong on a mud bottom. So I thought, well, maybe there was an increase in rainfall, but looking at tree ring records and again, other things, just couldn't find any change in frequency or magnitude, no change in El Niños or in the Pacific Decadal Oscillation, just can't find any change in climate from independent means. It correlates with the timing. Thought maybe it was all that concreting of the LA River, but that didn't start until the 1950s. So then I sort of slapped my head. I mean, this needed to be a 19th century stressor, not 20th century. That's too late. So I started then throwing myself into history of land use, because there was absolutely no fishing bottom take going on the Southern California shelf. And it turns out in the 19th century, there was a lot of livestock. I mean, a lot of livestock. The Spanish introduced these livestock in 1771. This was the industry. This was not for beef, this was for leather. And then I started looking in the agronomy literature for sediment yields and found that unmanaged grazed lands, you have 10 times more solid sediment runoff than if you have a managed herds on grazed lands, and that water that runs off of a prairie without livestock is clear, that is zero. So I threw myself into quantifying this because I figured, well, if I could quantify the number of cows, then I could calculate the quantity of mud. And here's a plot I never thought I'd see, and much less produce. But this is the log number of cows and horses and everything else, which is the black line. And this is time on the bottom axis. 1771, when the Spaniards arrived and released the cattle. 1850, we start with US agricultural census data. So I use these two data sources. Believe me, I spent 5 weeks just throwing myself into this. And what you find is that that livestock went up exponentially. It's like microbes on a petri dish, because these were released. They did absolutely nothing to care for them or water, nothing. And then I found a report that in 1850, you needed 10 acres to support one cow calf. And we had 1 million acres of coastal plains, so that meant the carrying capacity was 100,000. So it appears that these are minimal estimates of number of animals. So I think capacity was probably reached by 1830. And the Spanish missionaries were culling feral horses by 1805 because there was so much competition for food. So I'm pretty sure that's what happened. So I think that's when the mud would have started to run off. And then with the statehood, all of that 1 million acres was converted by 1900, again, exponentially, to cultivation. But of course, this was before soil conservation. And you have even higher solid sediment runoff from poorly mismanaged agricultural lands. So I think we had fundamentally 100 years of elevated mud, peaking at around 9 megatons per year, which is about what you get from famously muddy rivers such as the Eel River. Now, the great thing is, still that was a correlation. The great thing is some guys in California went off and they cored off of Los Angeles and Santa Monica shelf. And although they weren't looking for it, the only change they could find in the last 450 years was in the mass accumulation rate of the sediment, with an apparent greater than 50% increase occurring between, 1850 and the early 1900s. And this constancy of sedimentation is because, of course, increasing urban agriculture and urban use, but increasing concreting and paving. So anyway, we didn't have to go and test this, but this is a first appearance of mud, not just in local estuaries and bays, but all across the continental shelf, out to the shelf slope break, and over 400 kilometers of California shelf. Okay, so now some biotaxomes. What do we know? What does this tell us? Biological, for working with our agencies. First, we can tell them that the Los Angeles shelf was a mosaic of habitats, including at least 3 different kinds of shell gravel communities. And Adam has gone in and dated the first 100 shells of these dead-only clammus scallops. And again, none of them are younger than 100 years. So we think virtually all of those filter feeders went out at the same time as the brachiopods. It was a complete demolition. Now, the mainland shelf back then, pre-Cal, did include muddy seabeds, because we see them on the shelves of the offshore Channel Islands, where Liqueus, the brachiopod, still thrives, but it's still sandy. There are muddy patches, though, but they have diverse array of feeders. They're not just detritivores. And so on the mainland shelf, back on the LA side of the Pacific, these widespread muds are fundamentally the new normal. And, we're talking about 20 centimeters of mud. The muds are healthier now than they were in the 1970s, when we had peak output of nutrients and solid sediment from wastewater. They're more diverse feeders than the biologists document in the 1970s. But they clearly have not yet recovered. They're on track, but they haven't recovered. And we know from our pre-Cal death assemblages that, for example, obligate deposit feeders should constitute around 20% of the diverse mud community, not just the 1 to 2 that we find today. Okay, so this was indeed a real ecosystem collapse. There's less habitat complexity now, less habitat, less seawater filtering, more detritus based, which means a less efficient nutrient cycling from an ecosystem perspective. And this was all before 20th century stressors. So some very, totally unsuspected, but very sobering insights. One, we had extirpation of a diverse ecosystem that had thrived for thousands of years in the middle and outer shelf. The loss was anthropogenic, but it was pre-urban. And it was not, well, you can call it industrial livestock, but it was pre-mechanization, completely unsuspected, despite 50 years of intense biomonitoring of the very same shelves. This loss was slow. It unfolded over 50 to 100 years. And so it's very sobering to think, if it were going on today, would we recognize it? It was initiated by solid sediment. And this is the first detection of solid sediment as a driver of ecosystem change on the open shelf, not coastal sinks, not lakes, on the open sort of continental shelf. And some workers have found a similar situation now on the North Sea, driven by medieval land use change. And finally, the recovery from the 20th century wastewater, we can tell them also that that's complete, but that the legacy mud is going to persist and is the new normal, placing a real premium on protecting these still sandy island shelves offshore. So I'm going to take my remaining 1 minute. Oh, wait, it took 5 minutes to introduce me, but let me show you a couple, get a little bit more provocative and what next. We want to move beyond, that's the thing about conservation paleobiology and historical analysis using fossil material, is that we are writing obituaries, increasingly refined obituaries. But the obituaries of lost taxa, lost communities, and increasingly lost ecosystems. Now, from live dead and age dating, just using the surface mixed layer, these super easily acquired dead shells, we can, however, learn about new anthropogenic stressors, such as in Los Angeles. And of course, we can reconstruct sequential effects of multiple stressors. And here, Rafael and Paulo and the whole group there in Vienna, and with Adam working in the Adriatic, some wonderful examples, a whole series of terrific talks, as well as many people working elsewhere. But these are really sort of these workers using this dual sort of approach. Importantly, this approach, we can exonerate stressors. We don't simply point fingers at humans. We can, in some instances, say, no, you didn't do it. For example, it wasn't wastewater, it was cows. We can establish also, of course, the spatial footprint of the effects of stressors because of the high habitat level spatial resolution of dead skeletal material. But we do need to start looking at more units of analysis, especially so we can connect with ecosystem scientists. They use biomass, not numerical abundance. And or they focus on the one or two sort of key taxa that are most important for transferring nutrient and energies from one level to another. And so this zooms us in to sort of target taxa as opposed to whole community. We need to assess changes and think in terms of what are the effects on ecosystem services, not the particular biodiversity, not the particular set of species, but the net effects, the consequences for water filtration, for natural community protection of shorelines, for food, sort of what we would call the bioeconomy. Sorry, that sort of hurts to think about our animals, but. And another thing is ocean acidification. My gosh, that is the top ranked item of all the agencies that we here in Chicago have been working with, especially on the West Coast of the US. But OA is going to be one of the most difficult things to extract from death assemblages. Brock Kokish is working on this right now in the lab. And that's because OA decreases shell input. Think of all those little bivalves. They're finding bivalves that don't have shells anymore when they're living, because the shell dissolves even while they're alive. And obviously then that increased sort of acidification can destroy the shells of past cohorts, right? That's this loss, absence of sequestration in the Arctic where it's already undersaturated overlying waters. So that then it actually sort of like an erosional unconformity, you not only not have new sediment accumulating when you're eroding, but that erosion is removing some of the sedimentary record you had previously accumulated. So this degrades the pre-OA shell record. This is just going to be a super tough nut to tackle, to solve. Finally, going down core, lots of more complicated, it's very complicated, and there's several of us now working on what happens with progressive burial. That's a whole other talk. But we can get historic range of variation just by age unmixing assemblages. And you need to age unmix even in core increments because of bioturbation. Those are still time averaged and prolonged time averaging. We also need to really seriously start integrating eDNA. If I were a graduate student now, I would learn eDNA, sedimentary DNA, that they are going to eat our lunch if we don't start, or we morphologists don't start working with them. And then finally, about the changes ahead, we need to start evaluating components of resilience and historic insights into sustainability. We need to refocus sort of our language and thinking. Recovery and restoration, we're really good at finding targets for that from our obituaries, but I do wonder, how many of those are realistic? We're not going to remove that mud on the mainland shelf. That's the new normal. And so there's been some real ratcheting. Invasive taxa can do the same thing. So again, step back, think about the structure and function of the community and the biota rather than its details, species level detail. On the other hand, and I know this is very provocative, whether you think positively or negatively, geoengineering is happening. It is going to be happening and within the next decade or so. And that is going to be tipping our temperatures backwards, solar reflectance, and also start slowing and eventually tipping back CO2. And so one thing we can really use is our quaternary records to simply envision the environments we want to live in, whatever they are. Okay, well, thank you again so much. Let me leave you with some smiling faces. Erin mentioned some of our kids. These are some of the first generation. There's Adam on the left. And here's some other Chicagoans, including Brock, who's the one student here at Chicago right now working on these level. Anyway, thank you so much for the invitation. It was a real honor, and I look forward to participating with you all for the remainder of your project.
Thank you so much, Sue. That was terrific talk. What an absolutely great way to kick off our seminar series, especially with that broad view perspective that you bring, and the future questions and perspectives. Wonderful. Thank you so much. So questions for Sue are open, so you can use the Q&A button on the bottom right. We have some questions already coming in, which I can read out. But if you prefer to read out your question or say your question yourself, please just press raise hand and I'll allow you to speak. No problem. So let's start with a question that came in from Corinna Myers. So she asks, hi Sue, I'm curious how you conceptually think about, actually this question kind of gets at the crux of your presentation. I'm curious how you conceptually think about, interpret fossil communities to the tens, to the thousands, to the millions of years, compared to how ecologists consider modern communities of the tens to 100 years. That is, what is a community when time averaged over geological versus modern timescales?
Great question. I'm writing it down because I want to remember to be more explicit about that in the future.
I'll put it in the chat so you can read it again, Sue.
Yeah, okay. Here's the thing. Hey, Corey, a great question. Exactly. What does it mean? The first thing I want to say is that time species relation, we have these things, species area relationships, right? How many species do you encounter as you sample 1 square meter, a hectare, more, and of course the number of species you encounter goes up, in part because you encounter more individuals. But even if you sample size standardize, you get more species because a big spatial footprint gives you an opportunity to sample more different subhabitats and variability. Okay, so we all accept that the species richness that you measure at a hectare is just as true as the species richness you measure in a square meter. It's just truth at a different spatial scale. And that is fundamentally my answer, first order answer about time scale. There is nothing untrue about the super high diversity we encounter in a death assemblage that's been time averaged over 1,000 years, as opposed to one time averaged over 10 years. It's simply true at a different scale. So one of the key things that would be important is sort of trying to establish or to learn some qualitative methods of estimating scales, order magnitude scales of time averaging in fossil assemblages, that is things we get from cores and outcrops, so that we know how to put this in perspective. Now, I don't know if I can go back in slides, but if you go back to that slide where Adam's my paper, I showed you the simulations that Adam had done. And what you may have noticed is that, for example, richness increased rapidly with scale of time averaging, but it then began to level off after a few hundred years or 500 years. And so that's good news, but that presumes steady state community over that period of time. And so that's the other complexity, is that when you increase the window of time, you're giving an opportunity for secular trends, natural trends in temperature and organic. And so fundamentally, I mean, so here is then the final answer from this, and that is that the biological information we get from assemblages that have prolonged time averaging, it's a fundamentally different kind of picture. Go back to the teenager's bedroom floor. It's a fundamentally different kind of picture than what a biologist would get, even when the biologist has a time series that they analytically sum coarsen the perspective some. And fundamentally, one level you'd want to be, comparing assemblages of similar scales of time averaging, as well as sort of settings. But this is that is indeed, one of the frontiers. Adam and I had at one point done some simulations of what happened to species composition over the course of marine transgression that our outer shelf sites today had experienced during their 10, 20,000 years of time averaging. So we allowed it to formerly have had shallow water living assemblages, and then middle shelf living assemblages, and then finally the outer shelf living assemblages that we sampled today. And because of the fading of the oldest, we found that, actually the death assemblage had the composition it should have, with progressive fading of the earlier phases. So that's one thing that does happen, is despite sort of prolonged time averaging, because most of the shells are from the last little bit, it sharpens. Oh, but Adam would be having me make one final correction. And that is one of the things we find when these surface death assemblages transit into the permanent record, is that you lose that you lose the L shape. The age frequency distribution flattens. And so we have a more even distribution, more uniform distribution of representation of individuals from each of the cohorts. So that is the modeling we have not yet done, but Adam, I'm sure, is taking notes if he's here. And we will, because we need to go ahead and publish that work. But now we know how to make it completely more relevant to the deeper time fossil record. Sorry, I only answer in 4-paragraph answers. Great question, thank you.
Thanks, Sue. So we have 3 questions that people would like to ask themselves. So up first is Daniela. Daniela, can you turn your microphone on and ask your question?
Hi, can you hear me?
Yes, yes.
Thank you very much for the really interesting talk. We have in the past used CT scanning to look at bivalves, historical, so 100 years old bivalves in the Southern Ocean. And I was wondering if you had considered the potential of the methodology allowing us to look at density, surface to volume, because you talked about biomass, as well as internal dissolution in the shell, which can tell us something about some of the pressures the organisms might have been under, i.e. to link from your observation that the siltation threatened it, to considering what the physiological impacts were.
Yeah, that would be really great. And yes, I'm actually fairly optimistic of that. But you probably get, though, I'm a fundamentally sort of cheerful and first order, arm's length sort of person. But I'm fundamentally optimistic about that. And that's because some of the work I didn't show you, I cut out a whole segment that was just too in the weeds about time averaging. And one of the things I'm really interested in as a geologist is why and how those aragonite shells actually persist as long as they do. What they were just lucky, or did they undergo diagenetic stabilization? And we know both from our modeling that they have to have slowed their rate of dissolution in order to survive reworking and persistence. But I now have a lot of SEM data from tropical, temperate, and Arctic sort of settings. And what I find, this GSA talk from the last couple of years, and I hope to write this up this summer, is that in all 3 sites, we do have stabilization. The stabilization, though, takes the form of syntaxial cementation of new carbonate. And I'm just looking on the inner shell surfaces so that I know that, they started out right. They didn't have any post-mortem damage to the external, just looking at the internal surface, like just as you mentioned. And so it's a superficial skim, just, sort of micron, a few micron scale. But the great thing is it's superficial rather than penetrative diagenesis. And it's not even just modifying this existing surface, it's additive. It's a skim on top of, applied to. Now, what I don't know, and this is one thing we're doing this summer, is use Raman and figure out, is it aragonite or is it calcite? It's possible it's calcite. But even if it's just aragonite, these newly deposited crystals, and it's definitely microbial, okay, are coarser than the microcrystallites of the shell structure, the molluscan shell structure. And so they should be less reactive simply because they have less surface area per mass ratio. Now, what that means for your question is that, in a sense, I think it's sealing in all those growth lines that, people have known for a long time are preserved, right, in the fossil record. This explains how they sort of get survived. Now, that could be bad news, however, though, in being able to still detect physiological damage, because we'd have to sort of strip that surface skim off. Now, on the other side, the positive side, is that those skims, the latitudinal trend we find is that those skims appear within the first couple decades postmortem in Southern California. And I have yet to do the calibration, but they arise at least as fast, and I think faster in the tropics, because I find them more broadly across the available shells. But in the Arctic, we have only found those superficial skirms appearing on our oldest shells. And so I think that's in part because it's microbial and it's cold water and everything is slowed down. And maybe it's also tougher to get calcium carbonate precipitated, even if you're a microbe and really micro environment. But what that means, though, is that there be more unskim coated dead shells in high latitude settings. Anyway, that's how I'm thinking through. I'm fundamentally optimistic about a bunch of stuff. I am really concerned about this OA thing, including, as you said, the physiologic response. And in part, that's because, of course, post-mortem things also go right at that inner shell surface. Is that sufficient?
Thanks, Sue. Daniela, do you want to add anything?
Thank you very much. I think I'll reach out in an email and show some images.
Yeah, that'd be great. That'd be great. I want to see baby pictures. I think many of us have SEMs that look just like the ones we've been looking at from a taphonomic perspective, the geologists especially. But it would be great to see some from someone with a more biological take on the whole problem. That would be great. Thank you.
Sue, we're at 11:06. I hope it's okay if we can, if you have time to ask a couple more questions. If you have to leave,
sure,
then no problem.
Sure, I can stay, no problem.
Thanks so much.
I have a faculty meeting in an hour and a half, but we'll run out of questions before then.
Thanks a lot. So Amy would like to ask a question. Amy, could you turn on your microphone and ask away?
Yeah, hi, can you hear me?
Yes.
Hi, so thank you for your talk. I found it to be very compelling in so many different ways, and I hope Aaron has been recording it, because I could foresee somebody listening to this talk over and over again and pulling out different things, So just real quickly, I have to preface this with a little bit of background. I'm an archaeologist and a paleontologist, did my undergraduate work at Beloit College down the road from you. And I'm sitting in the foothills of the San Gabriel Mountains. So it was really serendipitous that I decided to listen to this talk today that's right in my backyard.
Yes.
So to speak. So, there's so much there that my head's just spinning. And then also I finished my work, my PhD in 1993 in Minnesota as part of the Limnological Research Center. So my question is about your dating of the recent sedimentation, the 1870s to the 19-teens. Coming out of Minnesota, we use glacial varves to do annual, or very fine scale resolution dating. How were these sediments dated here offshore?
Okay, so first off, the age dating resolution, the decadal age resolution that we show for the shells themselves, that is the resolving power of amino acid rascalization.
Oh, okay.
Which got a really bad rap because when they were first working on it, first trying this effort, I suppose back in the 70s, they were working on land snails. And it turns out there's a very strong thermal effect. But if you're working with shells, and Daryl Kaufman is the person that I and many of us sort of work with there in Northern Arizona, if you're working sort of subtidal, then you don't have all the wild temperature variations post-mortem. Now, rascalization goes faster in the tropics than in the temperate, than in the Arctic. So by decadal, but by but decadal resolution in temperate does mean basically decadal. And so that plot that Adam had sort of reconstructed the living abundance, the error bars on the timing of the decline in these brachiopod populations allowed for the uncertainty in the age dating method, the amino acid rasmusation. Now, when you get up to the Arctic, decadal means is a Kidwellian version of decadal, which ranges from 5 to 50 years. And decadal is more like 50 years or worse. In fact, we're going straight to radiocarbon in the Arctic now. Now, in terms of that core offshore, the UCLA, USC crowd, I'm not exactly sure. You know, they got so many things to age model. They were, but it was from slope water depths, which is why I didn't want to get involved in doing it. To launch, you know, it's a whole different, a whole bunch of things to tool up on. But, you know, there are all kinds of anthropogenic markers that they can be using, like pollen, the first appearance of eucalyptus and whatever. But I think they were also using VARV counting. So they had, and as well as lead-210 and whatever. So that's I got that resolution. We can't. We are when on the shelf. Adam and I, of course, have cores from 50 and 70 meters on the continental shelf. But by turbation, you know, the surface mixed layer is 20 to 40 centimeters, the surface mixed layer with age homogeneous lead-210. So needless to say, you know, we've got a lot of sort of bioturbational mixing, and we're not going to be able to resolve aging using the sedimentary matrix. The slope and rise, absolutely. So these guys went exactly where it needed to happen, which is why I'm so happy they found our mud.
Yeah, so I mean, what I'm getting at, as now I'm putting the archaeology hat on here, there seems to be sort of a discrepancy in the chronology. I find it a less compelling argument that the livestock signal is what you're picking up than railroads appear in the 1870s and 80s in the LA area, and the citrus industry took off in 1870. Yep. And I can put a link in the chat for you. UC Davis did a publication on the history of the citrus industry. So kind of consider that. I mean, 1848 was the treaty with Mexico. So maybe there's a time lag in your dating, you know, that you're picking up the signal later, or maybe there's another causation.
Yeah, you know, I'm from Virginia and grew up in a city. I'm not a cowgirl. And I'm not a Southern California girl. And so I broached this completely like, what the hell is going on? I threw myself into this for 5 weeks, using Bancroft and whatever. I can bore you to tears. I direct you to the supplemental online material of my thing. The cattle work insane. Railroads, absolutely, but railroads come in and they make one cut. There's nothing fundamentally erosive, you know, thinking as a geologist, how much land surface is affected. Citrus absolutely sort of came in but of course there had been dry lands, farming of wheat. So I took this apart, including then going to the agronomy sort of literature. When I first thought sort of like, my gosh, what was going on? I went and talked with one of the LA County sanitation biologists. It's wonderful working with municipal agencies because most of these guys, they live locally. They're long-term residents. They really know a lot about their region and they care about it. And I told Don Don Cadian, what we were finding with the brachiopods and that they seem to have gone extinct. And I said, because I knew he had seen their dead shells because he had picked through so many samples. And I said, do you think, I said, the only thing I can find that existed in the 19th century, deep in the 19th century, is livestock. I said, would there possibly been enough? And he said, there were insane numbers of livestock. And then I said, so what, they like overgrazed and then so there wasn't the plant life to take up the water? He said, no. He said, the thing about cows, it's not that, and livestock, it's not that they destroy the vegetation. It's that they trample the ground. They compact the ground, which reduces infiltration by rain. So even with no change in rainfall rate, a greater proportion of rainfall runs as surface flow. That then spoke to my geologist's heart. You need surface flow to transport sediment. And so I've also looked at the possibility of nitrogen from the cows, because there were 100,000 cows out there for a while. That's a lot of cow plops. I won't go into that, but I'm convinced that it's a solid sediment. We definitely did have solid sediment. And I think these guys who have the really well-dated core from Santa Monica Slope, that the 50% increase was around 1850. And they have really good chronology. And in 1850, that's pre-citrus, that's pre-statehood, that's pre-agriculture, that's pre-railroad. And the thing is, it takes a while. You have to move the sediment. It takes finite time. This thing you learn as a geomorphologist, right?
Right.
And even, and we're talking about a sandy, shelly seabed across the entire continental shelf that became muddy, not muddish, but was transformed to being a muddy seabed, not muddy sand, but sandy mud and mud. And that is a lot of mud, and it takes a lot of quantity, and it takes a lot of years. So yeah, I've gotten into the counterpart of the sediment delivery and river runoff, and this is a real cross ecosystem effect. I mean, these are cows on land affecting brachiopods and other filter feeders on the open continental shelf. So it's sort of massive. So I've got, as Adam knows, I've got a long, lovingly detailed, report just on that one plot, those plots of the cultural sort of history that's environmentally significant that I need to finish and submit. Because I think when the guys in the North Sea, the North Germans sort of found some similar things, not as far offshore affecting oyster reefs, it's still fairly near shore. The spatial footprint isn't yet clear, but it's sort of like, yes, okay. The sediment was really escaping. All of us know, I mean, the effect of damming, and for those of you doing quaternary, Savitsky did a nice paper in Journal of Geology probably 10 years ago or so now, because of course he's a big name in Anthropocene and sediment runoff and whatever. And he found that, a lot of what we observed sediment starvation on many continental shelves, he thinks it's a prolongation of early sort of transgressive conditions because of damming. Now, this would be, for example, along the east coast of the US. Beavers had been damming and holding back sediment runoff. But then, of course, they were extirpated by the hunters. But then, yes, farmers came in, but then they started wanting to control water, to feed their livestock, to control floods, to power mills, water mills, right? Water power, early industrial. And of course, each one of those gazillions of little dams is a sediment trap that keeps sediment up in the watershed. So he felt, and I want to go back and reread that paper, that it was a substantive starvation of siliciclastics, failure to reach the continental shelf. This is the exact opposite in Southern California. You had a few settlers and a gazillion cows, and they exploded exponentially.
Yeah,
Hey,
I think that your chronology fits.
Next time I'm out in the San Gabriels, that would be great.
Yeah. I think your chronology fits really well as a great mirror of the urbanization process that occurred from the 1870s to the 19-teens. And cows certainly were an important part of that mixture, and only intensified with the increase of population as well. Human population.
But they have to be free range, unmanaged cattle. No supplemental forage, no nothing. So they're all over the place. And the thing is that agriculture took off, but it started in 1850, and it took 50 years before it encompassed the same amount, the same totality of the watershed that the livestock were on. So I think, the one sediment source handed over to the other sediment source, two different drivers. And the effect of urbanization, all the agronomy literature, the effect of urbanization is to decrease sediment runoff. You increase chemicals, right? Dissolved load, petrochemicals, contaminants, DDT, but you decrease sediment. Too many suburban lawns, too much pavement, and of course you control runoff. And now they have almost complete reclamation of stormwater. So there's almost no water, even out of the pipe openings of these wastewater. They reclaim, it's used to water golf courses and things like that. It's urbanization itself, with buildings and people and factories and government buildings and well-managed railroads and roads, that decreases sediment runoff. The shelf is now fully starved out there. Okay.
Thank you. Thanks, Amy, for the question. That was great. I could listen to this all day. Sue, I think one of the great things that you mentioned there, or kind of showed us there, is that we can all take back to our systems is the local understanding of the local systems and the local history. And that's something that I think we can all learn from. That's great. We have, if it's okay with you, Sue, we have one more question from John Cybulski to ask.
Yeah, sure. Yeah, sure.
Great.
I want to put in one pitch for local and regional. Absolutely. And this is one of the things I most love about what we have discovered from live dead studies and all the taphonomic work. And we started discovering it from the very first live deads back in the 50s, which were motivated by oil exploration. They worried about blurring the paleoecologic evidence of facies. And we keep finding in subtidal level bottom settings, not washover fans, not continental slopes with turbidites, but level bottom shelves. There's very little postmortem transport out of habitat. And what that means, I think all of conservation, I think restoration, all that stuff, what we need are regional studies, regional sort of solutions. It's where we get political will, it's where we can have enforcement, we can get, will for regulations and benefits and economic, hiring people and all those sorts of things. So this has just worked out beautifully for us to really use this tool where it matters for us to save the world. Yeah, but one more science.
John, go ahead with your question.
John, great.
Awesome. Thanks. Thank you, Sue. I agree with Aaron. I could just listen to you kind of chat about cows and taphonomy all day. So if you want to skip your meeting, you could just continue, please.
Oh, I have a slide deck I could put up. Yeah.
Well, we could chat. No, I totally agree about the regional studies too. And actually, my question is sort of in line with that. And it's, the field is now growing. It encompasses people like me. I consider myself a historical ecologist, paleobiologist, conservation paleobiology, and more studies, which I think is great, are expanding the taxa or the fossils that they are analyzing within their records, either down core or live dead. It's not just shells anymore, but we're spanning, I would almost say, a size range of these actual preserved fossils. So from, I've seen studies looking at both, 4am shells and corals all in the same core. And my question is just about your life's work and all the trends that you see in fidelity, resolution, and time averaging, and what your feeling is about how that plays out across these taxa. So for these studies, and as we continue to diversify what we're looking at, if we date the shells or we date the coral, what do you think what can that be said about the other taxa that we might also be investigating? Because in my head, I imagine, that time averaging of small fossils is going to be vastly different. And I would think that they would, be more likely to be moved post-mortem, et cetera. And I don't know if there's any work you could point to that's looking at this specifically, or if you just have any general opinions, I would love to hear.
Yeah, all of the above. And here again, I'm going to show just what an optimist and a big scale. I hate microscopes. It's amazing I'm working on SEM at all, but I felt I needed to go to SEM to understand the mechanisms of that stabilization. So I've got an undergrad to help me this summer. Anyway, I think here's my first order take, that skeletal carbonate is skeletal carbonate. Okay. End of sentence. Okay. Now, it's the differences as a function of body size, mineralogy, microstructure, which, for example, includes the percent organic matrix mixed in with the mineral. Those things are degree. All of these things have significant prolonged time averaging. Now, one of the things in simplifying this talk so I could cover this much, and thank you for letting me just have these questions go on so long. is the very first slide I showed with Keith Meldahl's and Carl Fless's sort of age frequency distribution for Keone bivalves in Baja consumption bed. They also had this kind of data for the intertidal flats up at Choya Bay, just the other side of the Gulf of California. And they found the same scale time averaging. In fact, they had bivalve shells there that were up to 3,000 years. And we commonly find 3,000-year-old shells on beaches. And that's because all these beach positions, these intertidal flats, have been, the shorelines today have fundamentally been in the same place for the last 5,000 or 6,000 years when sea level rise reached stillstand. So, our geomorphology is basically the same. So again, a few thousand years, shells still kicking around despite bioturbation, despite aggradation of the flats, all that kind of stuff. Now, Ron Martin, University of Delaware, went in and age-dated forams from that same intertidal flat. And he found the same scale of time averaging. He found forams up to several thousand years old. And all along, one of the sources I always had, just way back, before I ever did anything out in the modern environment, is the literature on the radiocarbon ages of coral tops in the deep sea. And they would commonly, they'd date, it'd be a bulk calcium carbonate, but it'd largely be sort of forams from the top centimeter or whatever. And they were regularly getting ages that were several thousand years old. And so it's like, well, maybe we blew off when the core went in, and maybe we blew off and we actually lost the sediment water interface, blah, blah, blah. No. And even they came to realize, no, it's scale of time averaging. Now, the ODP people call this age offset. So when Adam and I published their first big core-based paper from Southern California in AGU's Paleoceanography Journal, we had to translate everything. We had to shift to talking about age offsets instead of time averaging. But there is a huge literature on just these kinds of issues in the deep sea. And to me, it simply sort of confirms forams, although they're tiny, they're only sand-sized. Of course, I think they're calcite. But the thing is, I think they're also pretty low organic. Now, we have, Adam took these age frequency distributions for Liqueus Brachiopods, which are calcitic, to sort of compare those initial loss rates to get our first order, our lambda 1 versus the lambda 2. And what he found was that the initial loss rates still much fast. They had the same L shape. You saw that. They're same L shape. But the rate of loss during that initial phase was around, I think, and Adam can correct me, but it was around twice, only twice as fast as for aragonite. That is, the calcite shells were disappearing 2 times faster than the aragonite guys sitting next to them. So that, for example, is an example of a mineralogic effect. Now, I know your reaction is going to be, oh my gosh, it's twice as fast. That actually isn't enough of a difference, though, to create to have those calcitic things that much more abundant. You simply can't, when you run the numbers, there are a lot of funny things that run against your intuition, but it's not sufficient to push the brachiopods up into, a high rank in the death assemblage compared to the aragonite. It's just like when people say, oh, if you count both valves of a bivalve, when you find them disarticulated, if you count all of those as rather than just the left valves, then you're doubling the number of bivalves and you're inflating their proportional abundance. Yes, but not as much as you think, because if you count every valve, both the lefts and rights, that also increases the size of the denominator. It doesn't just affect the numerator. These are these things you don't think about until you sit down and put it on paper and walk through and realize, oh, no. I'm doubling the numerator for bivalves, but I'm also doubling the numerator for the denominator for the bivalves. And yes, there are some snails there, but most things are bivalves. Most species are snails, but most individuals are bivalves. So the thing is, yes, it does some inflation, but not that much. And it doesn't flip things wildly in rank order. They may switch from first to second, a little switch of adjacent species in a rank order. So some of these counting things, anyway, I think it's turning out it's the same sort of thing. Now, Pelo Albano and others there at Veen, did wonderful,
Yeah, yeah.
published, right? And they dated the fish otolith, which are arachnitic, alongside the
Mhm.
bivalves. And what did they find, John?
Yeah, similar.
Of time averaging. Now, John Pandolfi's work back, when he dated sort of multiple coral heads and single horizons, and he did that for multiple horizons. And they were finding, my recollection is they found a smaller degree of time averaging there, but it was still multiple hundreds. But I do, so who knows, right? I mean, in tropical worlds, it's a different world. You notice I stayed with those plastics because they're simple to understand them. It's easy to differentiate matrix and shell when you're gutless plastic.
Yeah, I was lured into the tropical world early in my science career, and now I realize I'm like, oof, maybe I should have been farther north.
No, don't regret it. It's a good springboard, but yeah. But I think many of the other dynamics, all the modeling that we've done, the effects of time averaging, that applies whether you're talking about pollen, whether you're talking about leaf litter, whether you're talking about mammal bones or whatever. And of course, Josh Miller and Becca Turry have both done radiocarbon dating on large and small mammals. And again, I think this L shape is just what things look like, and it's remarkable how long things can persist. It's an L-shaped frequency distribution, because there is a lot of loss. And the other thing that I think we're finding when we go down core, that you lose that first thing. Fundamentally, once you move a death assemblage below the surface mixed layer, it can no longer be refreshed with newly dead shells. And so the rapid rate of loss, that first bar just rapidly collapsed. It's not being replenished with newly dead. And so that collapses. I think that's probably also, and Mike Kovalevsky and Daniel Scarponi did a wonderful, they went down core, Holocene core, and age dated, where they had assemblages. And again, you tended to have, flatter frequency distributions, and also, fitting with sedimentation rate being the first order control and time averaging, they had a broader range of ages of skeletal material at the surface of maximum transgression with the maximum likely duration of hiatus than they did at individual flooding surfaces. So it's all fitting together. And I think, as an order of magnitude type person and as a rank order rather than a picky sort of proportional abundance person, I think there's, the thing is to keep your eye on the forest. There are going to be details in degrees, but I think fundamental, some of these dynamics are just general to time averaging and to the potential for very early diagenetic stabilization, which of course could also be happening with some organic materials. I think, integrating ourselves with more organic stuff and especially eDNA, man, that is really going to transform things, because we can tell the world things that an eDNA person can't, sedimentary DNA person can't. But obviously they can get at groups and whole components that we can't. So we need to link. Shallow marine is going to be super, super tough place to capture that, but you guys
awesome. I appreciate it. I've kind of been waiting to ask you that question in person for a long time, so I'm glad that I got the chance. But it's encouraging to hear that, once you're below the surface, we're kind of working with that, as you mentioned, that 1%. But it seems to be pretty. The fidelity seems to be pretty good.
I think, Adam, we need to do the next level of that model, Adam. What then happens when you collapse? Because it is important, and people want to know. But but the scales of time averaging are scary long in temperate and tropical shelves, and that's because of just how durable those darned aragonitic and calcic shells are. And of course, the bioturbation with all the potential to exhume them back up and pump others down. And so you have very low temporal distinctness of successive increments. That's the other thing to remember, right? You may subdivide, you may subsample by centimeter, but you're fooling yourself if that does anything for your temporal resolution. Because if the mixed layer is 10, which is canonical, or 8, but is in reality more like 20, or more
Mhm.
on shelves and, can be comparable, greater in lagoons and that kind of stuff, then that a mixed layer means age homogeneity. It means super young things at the bottom and super old things mixed at the top. Now, you can subdivide it as finely as you want, but it's not going to get you anywhere historically unless you unmix the increments, which is what we're doing in Southern California, and Adam and Martin and the others in the Adriatic are doing the same to immense power. They're way ahead of Southern California in terms of actually turning that into an understanding of ecologic history and environmental. So I, just go read every single paper out of University of Vienna. And I apologize. I'm such a solistoclastic person that there are other really great groups going out there.
No, that's fine. That's fantastic. I appreciate it because, yeah, you're right. I'm working in the tropics and often with cores and often with age models created from, dating corals, et cetera. So it's just interesting. It's good to hear you talk about it, and I appreciate your time.
Sure, my pleasure.
Thanks for the question, John, and thanks for answering them so beautifully, Susan. That was wonderful.
Pages at a time. Oh, you're the right group for me to be with.
I think you found us. We found you. So before we leave, and thank Sue again, I just want to mention that, let me just put this forward. We have a pretty neat lineup of people coming for the Q-Mare seminar series for the next year and a little bit. A couple of spots are available. And next month we have Nicholas Piensen. So we're shifting gear completely, but with similar kind of questions in some regard. So Nick is going to be talking about the origins and fates of ocean giants, from digital ichthyosaurs to spying on whales, and we're really looking forward to that. So that's on the 1st of June. The seminar series will be every Wednesday, more or less around the same time, depending on the time zone of the presenter. So please, keep an eye out on the webpage. Twitter's the place to get updates. And we look forward to entertaining you over the next couple of years or so with this seminar series. We hope it's productive and we hope you enjoy it very much. Thank you all again. And thank you, Susan. That was absolutely terrific. I can't think of a better person and a better subject to start off with on this seminar series. So, and thanks again. And to Konstantina, thank you so much for organizing this working group and building the community in this way. It's exactly what we need to do. So you want to say some words, Konstantina?
A big clap for you, Aaron, because you're managing this fantastically. And thank you, Susan. And I really hope we see you again. We have a lot of meetings also planned in the framework of Q-Mare, and we'll keep you updated.
Thanks.
Yeah.
Okay, bye-bye.